Welcome to another lecture on electron microscopy. I am Nonappa, Associate Professor at Tampere University. I believe in open education and openly sharing scientific knowledge. My goal is to make my educational content openly available for everyone. In my previous lectures, I have discussed about cryogenic transmission electron microscopy and electron tomography. Today, we are going to look into an open source tool called a virtual scanning electron microscopy simulator. Electron microscopy has revolutionized numerous areas of science. It has provided scientists with some of the intricate details at nanoscale of various objects. Like any other electron microscope, scanning electron microscope also comes with certain limitations. It's expensive, it requires skilled operators and also laboratory conditions. This also makes many students, teachers, and researchers inaccessible to the instrument. In this regard, virtual microscope simulators are powerful tools to overcome these barriers. It has the ability to empower, enhance, and encourage learning, teaching, and training. These freely available resources will make both the students and the teachers' life a lot easier. Many of you might have studied, some of you might have used SEM, but some of you have never seen SEM but only have theoretical knowledge. Irrespective of whether you are a beginner, an expert, a teacher, or student, or lifelong learner, this simple tool will help you to understand how scanning electron microscopy functions and how we can get a good image. This virtual microscope explorer tool is free and available for everyone, made by Microscopy Australia and Thermo Fisher Scientific. In the outline of this looks as shown here, and what you can do is to really start, you can just choose a sample. If you press choose sample, it gives a set of examples available in the library. You can look at some arthropods, everyday things that you use in daily activities and life, some forensic materials and hydrophobic surfaces, water repelling surfaces. For today's experiment, we can just go and look into one of those beautiful butterfly wings and how it looks like. You can select this sample. Once you select this sample, in the right side, you will see the diagram or ray diagram of an scanning electron microscope. And what you see here is it actually venting. Venting means it's filling air. Now you can load your sample. And as you see here, you have a sample stub. Over that, your sample is loaded. And the whole instrument setting shows a simple design, the cross-sectional view. Here we have a source or for electron gun. You have a lenses, and then you have certain detector here. And this is the chamber, sample chamber. Now, once you loaded your sample, you go and evacuate. And as you saw, now it is under vacuum. Now you can select some of the accelerating voltage. It depends on the sample and the sensitivity of the samples. Let us choose 10 kilo electron volt. And now we can also select the spot size, either 5 to 10 nanometer. And what we have to select is the Z height, how close this sample is. And once you have selected all this, you switch on the high voltage. And what you see is that electron beam now go through all these condenser lenses, and your sample is irradiating the sample, and you have a detector here. And what we see in the background is already our loaded sample, which is a butterfly wing. Now you can control the brightness and contrast focus by using these knobs. By choosing the brightness and control, you can get very clear image. Let us look into the correct focus. Maybe it is correct brightness and contrast. Now you have to focus it. As you can see, as you focus it, you see the background structures, and you can change the contrast, which is, so you can go a bit under focus or over focus and then move both way so that you can end up in the correct focus. Now you see the small scales here. It is still low magnification, so you can increase the magnification, more details, finer detail and smaller structure. As small structure gene, you can still change the contrast and focus to get better resolution. You can increase the magnification, you can increase further. Now you can play with the brightness and contrast, and you can see how increasing and changing the brightness and contrast affect how the structural details are visible. You can also change the focus. You can go slightly over focus or under focus, and then you can get the correct focus. You can go to really high magnification, and at this stage you start to see even finer details of butterfly wings. Again, you can change the brightness and contrast as the resolution increases. You can also change the focus, and then you have finally the correct image. What you can do now is that you can save this, you can print it, and you have the image. You can choose another sample if you want. You don't have to be the same. You can choose, say, some forensic materials such as hair. It can be cat hair. Let's take a cat hair, and you do the same process, and you get to see and do again the similar. You load the sample, then you evacuate, and you select the accelerating voltage. You select the spot size. You select the Z height, and you switch on the high voltage, and you see the cat hair here. Now you have to change the brightness, and by changing the brightness you can also change a bit focus here. You can increase the magnification. You see the finer structure on a cat hair, and you can change a bit with the magnification and also brightness and focus. We can go higher, and what we see the more finer details, and it can be, yes. So this is a very simple tool and very simple way to understand what actually happens when you change the voltage. What happens when I change the beam size now? Now something changes. And what happens if I also change the height? So now you have to go back and change the focus, maybe the brightness and the contrast. So you can play around this, and really you can also change the electron volt. What happens if I 20 kilovolt? Again, something changes. And then you have to again change the focus, brightness, contrast. Again, you can go for really large spot size. Now it completely becomes invisible or fuzzy. And then you have to change also with the focusing. You're approaching a better focus. So yeah, so it is possible you can play around this with the different spot size, accelerating voltage, a Z height, and then play around the focus. And this is a beautiful virtual SEM that you can learn how to get a better image, better resolution, and what is the correct parameter for real